Yeah, so hello. And in this video, this is the third video of this metabolism. So the third thing is our Krebs cycle, and it is actually easy. Okay, we know that thing in the early thing itself. So we are adding certain things to it. So this is seen in all aerobic organisms, and it is an oxidative decarboxylation. And the link reaction is also an oxidative decarboxylation because we actually have oxidation in four steps where NADH and FADH2 are produced. Okay, and also we have this decarboxylation in two steps where two carbon is produced. Okay, so that's it. Okay, yeah, and that is the thing which we have. Then, as it is known as oxidative decarboxylation, that it is known as amphibolic. I think it is the thing in a reaction in which ATP as well as NADH is produced. I think it is that. Then the so if you have a doubt, please refer for that. Okay, then it has 8 steps. Okay, and it is occurring in mitochondria, matrix in eukaryotes, and cytosol in prokaryotes. Okay, so yeah, that's it, and it is present in all. So, a spontaneous reaction generally means the delta G is negative, so that is it, the spontaneity. Okay, and also yes, that's it. Then this is actually the complete reaction, and in the reaction actually, uh, the acetyl CoA. So the acetyl CoA is actually transferred. Acetyl CoA is entering this cycle from the link reaction. So it is a two-carbon compound. So it actually combines with a four-carbon compound, oxaloacetate, and it forms our citrate or citric acid. Okay, whatever we call it, and we get this citrate in the six-carbon compound, and it is then converted to isocitrate, then alpha ketoglutarate, which is a five-carbon compound, and in this step we actually lose one carbon. Okay, so this is actually the first redox reaction of the process. Okay, and yes, and in this is actually the oxidative decarboxylation, and after and before the ketoglutaric acid we have this process, that is NADH is formed and CO2 is formed. So this occurs after and before this alpha ketoglutaric acid. Okay, isocitrate alpha ketoglutaric acid, then we produce succinate CoA, which is actually again the four carbon compound because a carbon dioxide is lost there. Okay, so only in these steps we are actually losing carbon dioxide. Okay, and from succinyl CoA we get succinate, and in that step we are actually getting an ATP or GTP. Okay, uh, in plants and animals it is different, ATP or GTP. Yes, so it is GTP in plants and ATP in plants and GTP in animals. Okay, mostly we are discussing that. Then from succinate fumarate is formed. In that step we are actually having FADH2. Okay, and in fumarate to malate there is no intermediate products. Okay, and this malate is converted to oxaloacetic acid, which is actually our the starting of the reaction itself. Okay, and in that step also one NADH is produced. Okay, so this is actually an overview about this Krebs cycle. Yes, and how we remember this is the CIK, then SSFMO, CIK SSFMO, and that is the Chunku is king. Okay, so switch FM on. Chunku is king, so switch FM on. Okay, that is our short form to remember this. How is it? Chunku is king. Okay, so switch FM on. Okay, so this is it, the 8 steps. This is citrate, isocitrate, ketoglutarate, then succinyl CoA. Okay, then succinate, then fumarate. Okay, malate, then oxaloacetate. Okay, so these are the steps which we are actually going through in the process. Okay, then yeah, so there we'll see in several steps and several reactions the enzymes synthetase and synthase. Okay, synthetase and synthase. Synthase are enzymes which actually can produce ATP and utilize ATP. Okay, so they can actually do forward reaction as well as backward reaction. Okay, but synthetases are the enzymes which actually only can produce ATP and not utilize ATP. Okay, so these are, they only do reaction in one direction. Okay, that is the this thing. Okay, synthase and synthetics. Yes, and the products from this reaction, okay, are in the 3 and 4th reaction, in the third and fourth reaction. So how we write these products is like, uh, we get this. Okay, we simply can write, it is like 3 4 5 6. Okay, then we skip 7 and 8. Okay, so that is 3 4 5 6, then 8. Okay, and after that, in 3 and 4 we get NADH. So we have seen this. Okay, in 3 and 4 we get NADH. Okay, and in 5 we get ATP or GTP, and in 6 we get FADH2. Okay, then in the 8th we again get NADH, and here we also get CO2. Okay, we also get CO2 in the third and fourth reaction. Okay, so this is how we do it. Okay, 3 4 5 6 and 8, and in the 3 and 4 we get NADH. Okay, NADH plus NADH plus CO2. Okay, then in a 5 we get ATP and GTP, then in 6 we get FADH2, and in 8 we get NADH. Okay, so this is the product of it, of the reactions. Yes, so now we are actually going to discuss these reactions in detail. Okay, before going to detail, one thing which we could remember is that why FADH2 is used here, not NADH. That is because see, FADH2 is actually focusing in the removal of carbon-carbon bond. Okay, in the oxidation of carbon-carbon bond. Okay, so FADH2 oxidize carbon-carbon, double bond and triple bond mainly. So we can actually say carbon-carbon bond. Okay, and NADH and the chain NADH mainly oxidize carbon oxygen bond. Okay, so as there is a carbon carbon bond, that's why it is actually doing. Okay, then also it is actually seen as NAD plus. Okay, this is actually the usual form which is actually present, which is again an oxidized form. Okay, and we can actually add two oxygen, and then only it becomes 2 electrons, then only it becomes NADH. Okay, but this is actually present as FADH, so that is actually a reduced form. So in order to take one electron, it should actually be becoming NADH. Okay, so yeah, that's it. So as it is an oxidized form, it can actually take NADH. Okay, but here it should be NADH2. Okay, so this is it. The reaction is actually NADH to FADH2, FADH2. Okay, and here it is NADH. Okay, so this is a funny factor and important factor. Okay, yeah, that's it. That's about that part. And yeah, also this ATP and GTP formation, we may discuss that later, I don't know. Yeah, so this GTP is formed in animals and ATP is formed in plants mostly. Okay, so yeah, that's it. We are discussing these steps like in detail. Yeah, so we were actually discussing about the citric acid cycle. So in these reactions, the oxaloacetic acid is first binding. Okay, oxaloacetic acid first binds to the acetyl CoA and forms the 6 carbon acid, that is the citric acid. Okay, so that is it. Then it is actually a prochiral molecule. That means it is not actually chiral, but it is showing chirality. Okay, so such molecule is known, or it is actually showing optical activity, and such molecule is known as prochiral molecule, and it is synthesized by citrate synthase, and this citrate synthase is actually an allosteric enzyme. So on binding of oxaloacetate, it actually shows structural changes for this acetyl CoA. Okay, so an allosteric enzyme. Okay, right. Then succinyl CoA is acting as inhibitor. ATP is acting as inhibitor. So long chain fatty acids. So except this long chain fatty acids, all the other products are like the products. Okay, so that's why they are also inhibitors, and these things are actually the substrates which are actually the promoters. Okay, then the citric acid is then converted to cis aconitase by aconitase enzyme. Okay, yeah, and this reaction is suppressed indirectly by fluoroacetate. Okay, so this is actually the step which we have written earlier, the citric acid to isocitrate. Okay, citrate to isocitrate, and it is actually done in two steps. It is citric acid is first converted to cis aconitase, then the cis aconitase is converted to isocitrate. Okay, yes, and now we have this both is done by this enzyme aconitase. Okay, and now we have isocitric acid. Okay, so isocitrate, this isocitrate is then converted to which one, alpha ketoglutaric acid, which is actually the first redox reaction, first decarboxylation reaction in this, and here actually we get NADH and CO2. Okay, so NADH and CO2 is released, that means it is an oxidative decarboxylation. And here also isocitrate dehydrogenase is acting, that is a which enzyme, that is a allosteric enzyme, that is an allosteric enzyme. Okay, so most of the times it is the substrate and the process which is happening here. Actually it is an oxidation, so substrate is isocitrate and the dehydrogenation, that's how we get the name of the enzyme. And sometimes we also name it as the product with the product. Okay, so that's it, we are going to see that. Okay, then the next reaction is this alpha ketoglutarate is taken and it is converted to succinyl CoA. So alpha ketoglutarate to succinyl CoA, and this is also and in this reaction also we get what, we get an CO2 and an NADH. Okay, CO2 plus NADH. Okay, so this is the reaction, and a CoA is added into the reaction. Okay, a CoA is added into the reaction, and this thing is known as alpha ketoglutarate dehydrogenase complex, which is actually having the E1, E2 and E3. നമ്മൾ മുന്നേ ഇങ്ങനെ ലിങ്ക് റിയാക്ഷനിൽ കണ്ട എൻസൈം പോലെ തന്നെ 3 കോംപ്ലക്സസ് ആണ് നമ്മുടെ, എനിക്കൊന്ന് ലിപ്പോയിൽ ഡീഹൈഡ്രോജനേസ്, ഡൈഹൈഡ്രോ ലിപ്പോയിൽ ഡീഹൈഡ്രോജനേസ്, അങ്ങനത്തെ 3 സാധനങ്ങൾ കൂടിയിട്ടാണ് ഈ കോയൻ്റെ ട്രാൻസ്ഫർ നടത്തുന്നത്. അതേപോലെ തന്നെ എന്ത് സംഭവിക്കും, സോ ആദ്യം വാട്ട് ദേ വിൽ ഡു ഈസ് ആക്ച്വലി ദേ വിൽ ടേക്ക് ഇറ്റ് ടു ദ TPP, ദ ഫസ്റ്റ് enzyme, the alpha ketoglutarate dehydrogenase. Okay, so it will actually take it the molecule to the TPP, and it will be transferred to the what I should say, lipolysine. Okay, then the lipo get reduced, and this reduced electron actually is transferred to NADH. So this reaction occurs here also, so as we have discussed in the link reaction. Okay, so that's the fourth step. Then in the fifth step, what happens is that the succinyl CoA is converted to succinate. Okay, succinyl CoA is converted to succinate, and this is carried out by the succinyl CoA synthetase. Okay, so yeah, the succinate synthetase. Okay, succinate synthase or synthetase, we have to make sure about that. So this is actually the succinyl CoA synthetase itself. Okay, synthetase, that means it can actually do forward and backward reactions, right? Okay, synthetase only produce ATP, no backward reaction. Okay, so that is it, synthetase, no backward reaction, so there we don't have any backward reactions. Okay, so yeah, that's it. So this is the step where succinyl CoA synthetase succinate synthetase is involved. Okay, or it is known as thiokinase. Okay, then in that step we get ATP and GTP. ATP is produced in plants and GTP is produced in animals. Okay, so mostly it is said as GTP. Yeah, then this succinate is actually converted to fumarate, and the enzyme is succinate dehydrogenase, or it is also known as fumarase. Succinase somewhere, I think. Then FADH2 is produced here. Why FADH2 is produced? Because As we have already discussed, this is actually a carbon carbon bond oxidation. That means that's why FADH2 is produced. Okay, so and this succinate dehydrogenase, the enzyme here, is actually used in the complex, used as the complex 2 in electron transport system. Okay, as the complex 2 in electron transport chain. Okay, and it is, yeah, that's it, and seen in inner mitochondrial membrane of eukaryotes and plasma membrane of prokaryotes. And malonate act as a competitive inhibitor of this. Okay, as a competitive inhibitor, it increases km and reduces the affinity. Okay, so it is due to the structural similarity of malonate to our succinate. Okay, then we have this ferrum and sulfur, which is acting as cofactor. Okay, then succinate. Okay, succinate, phosphate, അതേപോലെ ATP, then coenzyme QH2, QH2, all these are actually activating this enzyme. Okay, succinate is the actually the substrate, then ATP, then phosphate, all these things actually act as a what, act as a actually activator of this enzyme. Okay, then this fumarate is then converted to malate by malate synthase or the fumarase. Okay, so malate synthase or fumarase, yes, and malate is produced, and then this malate is converted to oxaloacetate by which one, oxaloacetate by um, malate dehydrogenase. Okay, malate dehydrogenase. Okay, so that's it. And in the process, in the citrate process, it is actually citrate synthase. Okay, it is actually citrate synthase. I have confirmed and verified this is actually citrate synthase here. And in case of this succinate, it is succinate synthetase. Okay, so this is a, this reaction cannot be reversed. Okay, this enzyme cannot utilize ATP. Yes, that is the thing. And this malate dehydrogenase, and here also an NADH is produced. Okay, yeah, we know the thing. And one thing is that this reaction is actually normally non-spontaneous. That is, the delta G is positive. Okay, but due to the need for NADH created by reducing oxaloacetate. Okay, so oxaloacetate is reduced that much. Oxaloacetate is utilized that much, and the inhibitor is actually lost. Okay, so we cannot actually say the need for NADH, whatever. The need for oxaloacetate is created by reducing it. Okay, and thus what happens, due to the pull, the need, this actually becomes -7. Okay, first plus 27 to -7 transition occurs due to that reduction in oxaloacetate. Okay, and thus this reaction becomes spontaneous. Okay, yeah, that's it. Then the energetics. So if we look at the energetics and what we have here, yeah, so Ca2 plus act as a negative positive regulator in most of the enzymes in the Krebs cycle. Okay, and ബാക്കി all mostly substrate product വെച്ചിട്ട് നമുക്ക് പറയാം, substrate will be activating and product will be repressing. Okay, so now the calculation is done as 1 ATP equal to 2.5, 1 NADH equal to 2.5 ATP, and 1 FADH2 equal to 1.5 ATP. Okay, so this is how now it is calculated. Okay, so what we actually get in the energetics is that from glycolysis, this are our products, 2 ATP and 2 NADPH, 2 ATP and 2 NADPH. And in link reaction, so in the glycolysis 2 pyruvate is formed, and this 2 pyruvate, that means these have to go through the cycle twice. Okay, so in these two pyruvates, okay, two pyruvates, and in the link reaction 1 NADPH is formed, and as there is 2 pyruvate, that is 2 NADPH. Okay, then in Krebs cycle also, in a single reaction we actually get 3 NADH, 1 FADH2 and 1 GTP. Okay, and as there are 2 cycles, we actually get how many things, that is 2 NADH, then 2 NADH in the link reaction, then the 6 NADH and 2 FADH2 and 2 ATP. Okay, so these are actually converted in the form of ATP, using this actually 1 NAD equal to 2.5 ATP and 1 FADH2 equal to 1.5 ATP, and we get actually 32 ATP in total. Okay, so just remember this, we get 6 NADH and 2 FADH2 and 2 ATP from Krebs cycle in total, and 2 NADH from this link reaction, and 2 NADH and 2 ATP from the glycolysis. So in total, glycolysis actually produced 7 uh, 7 ATP in total, and this Krebs cycle actually produced 15, 17 and 19 ATP in total. Okay, so adding all this, we get 32 ATP. Okay, so that is the thing. Then the next thing is the fate of carbon. Okay, this is actually simple, which we have already discussed in earlier sessions. And the fate of carbon simply means in the link reaction. So how we actually write the carbon? Okay, so from the glucose and becoming pyruvate. Okay, so in glucose there are actually 6 carbons. Okay, so let's write 1 2 3 4 5 and 6. Okay, 6 carbons are there in glucose. So the first carbon and 6th carbon is there, then the second carbon and 5th carbon, and third and 4th carbon. Okay, so these are the carbons which are actually present. Okay, so this first and 6th carbon, so let's write from the inside. This is actually the, in pyruvate there are actually only 3 carbons. Okay, that is the 1 2 and 3. Okay, 3 carbons are there, and the first carbon is actually from 3 and 4 of the glucose, and the second carbon is actually from 5 and 2 of glucose, and the third carbon is actually from this 1 and 6. Okay, 1 and 6 of glucose. Okay, so this is how this occurs the in case of pyruvate. Okay, so this is actually the 3 carbon compound, the pyruvate, which is actually produced after glycolysis. Okay, then in the link reaction, this carbon, that is the third carbon of the no, I think that is actually termed as the first carbon, I think, whatever, wait, yeah, not that carbon is removed. Okay, this carbon is removed. Okay, 3 and 4th carbon, that is the first carbon according to the pyruvate. Okay, so this carbon is actually removed in case of this, what, in case of our link reaction. Okay, so the third and 4th carbon, that is the inner carbons, are removed in the link reaction. Okay, yeah, that's it. So that's the first carbon of this thing, and the third and 4th of the glucose. Okay, that carbon is removed. Okay, and this thing enters the Krebs cycle. Okay, Krebs cycle, and in Krebs cycle no carbon from glucose is actually removed, but the 2 carbon dioxide removed are the carbon from the oxaloacetic acid, and those are the first and 4th carbon of the oxaloacetic acid is removed. Okay, so first and 4th carbon of oxaloacetic acid is removed, and 2 carbon is added from this molecule also. Okay, so that's how we get this thing. Okay, so how we say this 1 and 6, then 5 and 2, and here only there are 2, that is 2 steps are there. Okay, 2 rotations, 2 pyruvate is formed. So from pyruvate we do everything twice. Okay, because 2 times it should be done. Okay, so this is it, this is our Krebs cycle. It's actually simple, we know this earlier itself, so just got some additions, and that's it. Keep studying and do well. Yeah, that's it. Be strong.